I'd like to start by thanking Twist for the invitation to speak at the meeting today. So my name is Gavin Bendle and I'm Vice President of Research and Development at Neogene Therapeutics, a privately held preclinical stage biotechnology company that is focused on the development of novel T-cell based immunotherapies for the treatment of a broad spectrum of solid cancers. So this is our notice of forward-looking statements. And in my presentation today, I'm going to introduce to you our next generation, fully personalized engineered T-cell therapy that we're developing at Neogene Therapeutics. So the problem we're trying to solve is that effective T-cell therapy is still unavailable for most solid cancers. However, as you can see on the left-hand side of this slide, the curative potential of engineered T-cell therapies for the treatment of some hematological malignancies has clearly been demonstrated. And this type of T-cell therapy usually involves a patient's T-cells being genetically engineered with a tumor-reactive antigen receptor in an ex vivo manufacturing process. And these engineered T-cells are then reinfused back into the patient, where they can move through the body and seek out and destroy tumor cells. Now, the antigen receptors used in this process can either be a synthetic antigen receptor known as a chimeric antigen receptor, and this is referred to as a CAR, or the native antigen receptor expressed by T cells, the T cell receptor, and this is referred to as a TCR. However, a crucial bottleneck in extending this form of therapy beyond certain forms of blood cancers to patients with solid cancers has been a lack of access to truly tumor-specific antigen receptors, be those CARs or TCRs. Now, nevertheless, on the right-hand side of this slide, you can see that there is a form of adoptive T-cell therapy that does have proven curative potential for some solid cancer patients, and this is called TIL therapy. And TIL therapy involves the surgical excision of a tumor lesion, the isolation of viable tumor-infiltrating T cells, which are called TIL, the in vitro culture and expansion of these TIL, and their reinfusion in large numbers back into the patient. And we know that TIL can contain tumor-specific TCRs, but two major issues that has limited the broader application of TIL therapy is the requirement for surgical resection of a tumor lesion for access to the TIL, and their highly variable viability and expansion during the manufacturing process, and also their variable function after reinfusion back into the patient. Therefore, at Neogene, we aim to provide access to tumor-specific TIL-derived TCRs for engineered T-cell therapy of solid cancers. Now, at least part of the therapeutically active component in TIL therapy comprises of TCRs that are specific for tumor neoantigens, and this class of antigens represents potentially safe and effective targets for T-cell therapy for the following reasons. Neoantigens are potentially safe as they are derived from tumor-specific mutations, and thus they're not expressed in normal non-transformed cells, as this schematic on the left-hand side of this slide shows. Furthermore, as the schematic on the right-hand side of this slide shows, they can be strong and therefore effective T-cell antigens, as there is limited intrinsic immunological tolerance to this class of antigens, as they are absent during T-cell development and therefore are often seen as foreign by the immune system. However, the vast majority of tumor mutations, and therefore the tumor neoantigens to which they give rise to, are unique to a given patient and are not shared between patients. And this means that targeting this class of antigens in large numbers of patients requires a fully personalized approach. Now, I just mentioned that at least part of the therapeutically active component of TIL therapy comprises of neoantigen-specific T cells, and there is a body of evidence suggesting that it is T cell reactivity in the TIL product against neoantigens that drives the therapeutic response to this form of therapy. And on the left-hand side of this slide, you can see some examples from a number of publications documenting this association. Now, furthermore, on the right-hand side of this slide, this cartoon demonstrates that tumour material is highly enriched for neoantigen-specific T-cell receptors compared to peripheral blood. And that is why our neoantigen TCR isolation platform is focused on accessing neoantigen-specific TCRs directly from the tumour site rather than from peripheral blood. Now at Neogene, we're aiming to transform mutational profiles that are obtained as part of routine genomic testing of metastatic cancer patients into fully personalised engineered T-cell therapies. And this schematic provides a high-level overview of our fully personalised TCR gene therapy platform. So you can see that from a tumour biopsy, we can identify the sequences encoding a patient's mutation-derived neoantigens and the intratumoral TCR sequences from TIL that have infiltrated the tumour. Then, using our proprietary technology, which utilises a combination of synthetic biology tools and a functional genetic screening approach, we can identify which of the patient's tumour-derived TCRs are neoantigen-specific and use these TCRs to generate a multi-specificity neoantigen-specific TCR-engineered T-cell product with which we can then treat the patient with. Now, the Neogene TCR isolation platform, as I've just mentioned, utilizes synthetic biology tools as part of the process of identifying TCRs for therapeutic use. And we have a very fruitful collaboration with Twist Biosciences with respect to the development and supply of synthetic biology tools that are used in our TCR isolation process. So as I outlined on the previous slide, the process of TCR isolation at Neogene starts with targeted genomic sequencing of a tumour biopsy from a patient. And together with Twist, we use this sequencing information to generate synthetic DNA libraries of both intratumoral TCR pairs and mutation-derived neoantigens. Now at Neogene, we then use these Twist-generated libraries in a high-throughput functional genetic screen in which we screen reporter T cells expressing the TCR library against target cells expressing the neoantigen libraries in order to identify neoantigen-specific TCRs. Now, we believe that the use of synthetic biology tools in combination with our high-throughput functional genetic screening approach helps set us apart from others in this space with respect to our platform's breadth, sensitivity, and scalability. So with respect to breadth, we're able to access the broadest repertoire of neoantigen-specific TCRs, since our platform enables the identification of TCRs restricted to any one of the patient's HLA alleles, which are the most polymorphic genes in humans. So therefore, we can access TCRs that are restricted to either HLA class 1 or class 2 alleles. Now, with respect to sensitivity, the advantages of our platform are twofold. We can access the intratumoral TCR pool that is enriched in neoantigen-specific TCRs relative to peripheral blood. And furthermore, our genetic-based screening approach has very high sensitivity compared to conventional immunological-based screening approaches. Now, finally, with respect to scalability, we're utilizing synthetic components, and this replaces the need for viable patient T cells during the TCR isolation process. So this schematic on this slide provides a much more detailed overview of Neogene's neoantigen TCR isolation platform, and it also demonstrates that our process is based on limited biospecimen needs. And I'll walk you through the steps involved in this process because this is a somewhat complicated slide. So the first step involves the extraction of DNA and RNA from a tumour tissue, and also DNA extraction from PBMCs, which are derived from a small peripheral blood draw, which is also taken from the patient. And then in step 2, we use this information to sequence both the mutanome and the bulk unpaired TCR alpha and beta chains from a tumour. And it's important to remember here that the T-cell receptor is comprised of an alpha chain and a beta chain that together form the TCR heterodimer, and it is the TCR heterodimer that defines the specificity, the antigen specificity of a given TCR. Now in the third step, we use the bulk unpaired TCR sequencing data to synthesize a TCR library through combinatorial pairing of intratumoral TCR alpha and beta chain sequences. So as an example, if we take the 100 most prevalent TCR alpha chains found in the tumour and the 100 most prevalent TCR beta chains found in the tumour and combinatorially pair these, this results in a TCR library of 10,000 TCR variants. Or as another example, if we take the top 200 TCR alpha and beta chains found in the tumour and generate a combinatorially paired library with these, this would result in a library of 40,000 TCR variants, for example. Now in step 4, the synthesized TCR library is introduced into Jurkat reporter T cells by means of retroviral gene transfer. Now on the lower part of this slide, you can see that in parallel to generating the TCR library, we use the mutanome sequencing information to synthesize a mutation-derived tumor neoantigen library, or sets of libraries, I should say, and we introduce these into immortalized B cells that are also derived from the small peripheral blood draw that we take from the patient that I mentioned previously. So through this process, we can generate neoantigen expressing autologous B cells that function as antigen presenting cells in the functional genetic screen, or TCR library screen as we refer to it. And this TCR library screen is shown in steps 5 and step 6 of this schematic. So in step 5, we culture the TCR library expressing Jurkat reporter T cells together with the neoantigen expressing B cells in an overnight co-culture step. And then the next day, we isolate activated and non-activated T cell populations from these co-cultures, and then determine the neoantigen specific TCR alpha beta pairs from these reporter T cells using a next generation sequencing workflow that we've developed at Neogene, and which I will tell you more about in a couple of slides' time. Now finally, in step 7, we can then select which of the identified neoantigen specific TCRs that we want to use to generate the TCR engineered T cell product with which we will treat the patient with. Now the synthesis of TCR libraries through combinatorial pairing of TCRs is a key component of our TCR isolation process at Neogene. And it's important as it enables us to avoid using viable patient T cells in the TCR isolation process. So when generating these combinatorial TCR libraries, the desired outcome is the generation of high complexity libraries containing all possible TCR variants, being present multiple times in a narrower frequency range as possible, since this will maximize the sensitivity of our screening process and therefore obviously maximize our chances of identifying patient-specific neoantigen T-cell receptors. And what these graphs and the metrics below each graph show is that for 3 representative libraries synthesized by Twist, each of which comprised of the 100 most prevalent TCR alpha and the 100 most prevalent TCR beta chains from different colorectal cancer tumor samples. So here each TCR library comprised of 10,000 TCR variants. What these graphs and the metrics show is that all 10,000 TCR variants were present in each library, with each variant being present multiple times with relatively little variation in their representation. Since if you look at these graphs, which show the median number of times a TCR variant is present, you can see that the peak of each graph is to the right, and the peak is comparatively narrow, and in fact greater than 96% of the TCRs in each library fall within a fourfold range of the median. So it's clear that these high complexity synthetic TCR libraries are of a very high quality, and will enable us to maximize our chances of finding neoantigen specific TCRs in our functional genetic screening process. So having worked with Twist to be able to demonstrate that they can produce very high quality, high complexity synthetic TCR libraries, the next question that we really needed to address with respect to this TCR isolation platform was whether we can isolate TCRs with a defined antigen specificity from a high complexity TCR library with high sensitivity and high specificity. So to this end, Twist synthesized for us a TCR library by combinatorial pairing of 100 TCR alpha and 100 TCR beta chains, so a library comprising of 10,000 TCR variants, and 95 of the 100 TCR alpha chains and TCR beta chains in this library were tumor-derived TCRs of unknown specificity, but the remaining 5 TCR alpha and beta chains in the library were derived from 5 TCR alpha beta pairs with well-defined antigen specificities. And so our aim in this experiment was to ask if we could detect these 5 defined antigen specific TCRs amongst the 10,000 TCR variants using our functional genetic screening technology. And the genetic screening process involves the steps shown on this slide. So the TCR transduced Jurkat T cells are placed in an overnight co-culture with neoantigen library expressing B cells. And then following the overnight co-culture, the activated and non-activated TCR library transduced Jurkat T cell populations are separated. Genomic DNA is isolated from these two populations, and the TCR alpha beta pairs expressed by these TCR library transduced T cells are amplified. Now, these TCR amplicons are then fed into a next generation sequencing pipeline that identifies TCRs that are enriched in activated T cells and depleted in non-activated T cells. And thereby, this enables us to determine which of the TCRs in a given TCR library is actually neoantigen specific. So shown here is the results of this experiment that showed us that using the Neogene technology, we're able to successfully identify the 5 defined antigen specific TCRs within this TCR library containing 10,000 TCR variants. So let me explain how to interpret the figure shown on this slide. So each of the 10,000 TCRs shown in this figure is represented by a dot, with the 5 TCRs of a defined specificity being coloured dots, and the other 9,995 TCRs being coloured in black. And we've plotted the reactivity of each of the 10,000 TCRs in the presence of B cells engineered to express the cognate antigens recognized by the 5 defined antigen specific TCRs of the x-axis, and the reactivity of these same 10,000 TCRs in the presence of B cells that have not been engineered to express the cognate antigens on the y-axis. So TCRs that show no differential reactivity against either set of B cells will sit on or close to the red diagonal line, whilst T cells that show reactivity against the B cells engineered to express the cognate antigens recognized by the 5 defined antigen-specific TCRs in this library will sit to the right of the red line. And you can clearly see here that this plot shows that the 5 defined specific TCRs are distinguishable from the cloud of the other 9,995 TCRs as they sit well to the right of this cloud. So this experiment served to demonstrate the extremely high sensitivity and specificity of our technology, because all 5 of the specific TCRs were identified amongst the 10,000 TCR variants in the library, and also they were the most significant leads amongst all of these TCR variants in the library as well. So far in my presentation, I've shown you that Twist can provide us with high quality, high complexity TCR libraries, and that the high sensitivity and specificity of Neogene's TCR isolation platform enables the identification of antigen-specific TCRs from these high complexity TCR libraries. So I'm now going to discuss how we have validated our TCR isolation platform using tumour samples from patients with microsatellite stable colorectal carcinoma, which is also referred to as mismatched repair proficient colorectal carcinoma. Now, this form of colorectal cancer represents the great majority of colorectal cancer cases in both the US and Europe. Furthermore, it is a hard to treat cancer with extremely low response rates to immuno-oncology agents such as PD-1 pathway therapies. And this is partly because it has on average a very low mutational burden. As this figure on the left-hand side, which is taken from a New England Journal of Medicine article, demonstrates. So here the authors of this article have plotted the average mutational burden of different tumour types against the objective clinical response rate to anti-PD-1 or anti-PD-L1 antibody therapies. And so the objective response rate is on the y-axis, and the average number of coding somatic mutations is on the x-axis. And if you look at this figure, you can see that actually microsatellite stable colorectal carcinoma, which we've circled here in purple, actually has an even lower response rate to these agents than would be predicted based on its average mutational burden alone. So I think collectively what this demonstrates is that, you know, we were setting a very high bar for validation of the platform using this tumour type. Now on the right hand side, you can see that the 3 microsatellite stable colorectal carcinoma samples that we received from our collaborators at the Leiden University Medical Center here in the Netherlands all had the expected low mutational burden, and that in addition to this we also analysed an additional tumour which was a microsatellite instable colorectal carcinoma in this set of experiments that we also received from the LUMC. So this slide summarises the results of these patient screens, and these plots demonstrate that we were able to identify neoantigen specific TCRs, shown here as green dots in each of the plots, in all 3 of the analysed microsatellite stable colorectal carcinoma samples and in the microsatellite instable colorectal carcinoma tumour sample, which is shown in the plot on the right hand side. Furthermore, we could detect both HLA class 1 and class 2 TCRs restricted or specific for neoantigens, and this is important because we know that both class 1 and class 2 restricted T cell receptors can display tumour reactivity and can play an important role in anti-tumour immune responses. Furthermore, we were able to identify TCRs that were specific for different types of mutations, so either single amino acid mutations or frameshift mutations. And collectively, these data are very important as they demonstrate our ability to identify neoantigen-specific TCRs in low mutational burden cancers such as microsatellite stable colorectal carcinoma, and therefore point to the pan-cancer potential of our technology. So for the current generation of our fully personalized TCR therapy platform, we're accessing neoantigen-specific TCR genes from bespoke synthetic TCR libraries encoding patient-specific TCR sequences. So we generate unique TCR libraries for every single patient for which we carry out the TCR isolation process. However, a longer-term vision for our fully personalized TCR therapy platform involves us being able to access neoantigen-specific TCR genes from an off-the-shelf synthetic TCR library rather than from a patient-specific TCR library, as this would enable us to access therapeutic TCR genes with similar ease to modern antibody screening technologies. And therefore, to work towards this goal, we have formed a strategic collaboration with Twist to begin developing this approach. And this involves Twist creating a synthetic TCR library with maximal possible diversity, utilizing their best-in-class DNA synthesis capabilities, and Neogene designing a rational screening process for such a library based on our expertise in therapeutic TCR gene discovery. And we plan to first use the generated synthetic TCR library and screening technologies to develop TCR candidates against 2 undisclosed tumor targets. So in summary, I hope I've been able to show you over the last 30 minutes or so that, that we at Neogene have developed a novel paradigm-changing science, and that we're using this together with best-in-class synthetic biology tools supplied by Twist to identify safe and effective TIL-derived TCRs to enable fully personalized neoantigen-specific TCR therapy of solid cancers.